Okay, I think we can start now. We've got we're going to have a OGM Community of Interest presentation today. Myself, Dr. Eric McGinnis will be doing the moderating, as well as Susan is with us as well to help moderate. This is a regular Zoom meeting call, so I believe that everyone can participate. So if you do have questions, you can either put them in the chat if you don't want to talk yourself, but feel free to raise your hand and enter the discussion with us all here. So we have a relatively full agenda today of things we'd like to talk about. We're going to open today's meeting with a couple of cases, because usually we save those to the end and then we run out of time. So today we're going to do them first as a little change of pace. And if there's anybody else that has a case prepared and wants to present, just send a message in the chat in case we've missed you. And then we're going to move on and talk about some of the other items on the agenda, including the Canadian OGM Landscape Survey, which Eric is going to talk about. And then we're going to have some group discussion. We might actually group. I don't know, Eric, you can correct me if I'm wrong here, but maybe we'll group points 4 and 6, the group discussion experiences on Stratus and the open discussion, maybe together, and maybe we'll move the hypereosinophilia and OGM discussion up ahead of that. Just get that out of the way, because I think that's sort of a separate biological point as opposed to a sort of instrument technical issues. So if that's okay for everybody, I think that's where we'll start. Also, I will also say to everybody, I wanted to thank everybody for joining us today. This meeting is accredited, so you're going to get a survey. There's going to be a survey link in the chat at the end of the meeting. It's also going to be emailed after the meeting to everybody who signed up. So please fill in the survey. It's really important, and that also helps you get certificate for attendance if you need that for to add to your continuing education credits. Also, I think that's everything we needed to discuss. So I think we'll move into the cases. And so I would like to invite Nicholas to take over here from Vancouver General Hospital. And Nicholas, really thank you for offering to present a case today.
Okay, so my name is Nicholas. I work at Vancouver General Hospital with Dr. Eric McGinnis and Tara Spence, and today we'll be showing some of the interesting cases that we have seen from our OGM analysis. So, and we will be focusing on mostly mucin rearrangement that we have seen fairly interesting results and highlight basically those findings. So, the evaluated cohort that I included here was mostly cases that involve AMLs and other mild neoplasms. So we evaluated 147 cases between November 2023 and March 2025. As you can see from this diagram, most of them are AML. Some of them are acute undifferentiated leukemia and other high-grade mild neoplasms and therapy-related mild neoplasms. So the way that we process this is the typical workflow with OGM. Most of these specimens we process pretty fastly, and we have done like karyotype fish in some of these cases and OGM, and we basically compare how these findings correlate with each other. And as I mentioned, I focus here like on mostly cases harboring mucin rearrangement, and basically this diagram like represents most of these findings. So as you can see from your diagram here on the left side, we found like 5 cases that show abnormal karyotypes involving chromosome 3 in some way. We also find 2 additional cases that have a normal karyotype, but not necessarily involving that chromosome 3 harboring mucin. And we have 1 case with a normal karyotype and 1 case where we have a failed karyotype. And all these cases were also we will also do OGM on them, and what we noticed was like there were like multiple partners, not some of them the classical GATA2 inversions, some other also reported rearrangement involving TARA, and these 4 cases involving non-typical or not like as common partners. So I will show in detail what we see for all of these cases, and hopefully you can appreciate like how OGM is contributing to each one of them. So the first case that I'm showing is an exemplary case in a classical GATA2 MECOM rearrangement in chromosome 3. So basically what we have see here is a typical chromosome 3 inversion captured by karyotype, and we were able to see that by OGM as well. And this is what it's seen here fairly clearly in this circus plot. We see the inversion highlighted here in this pink ribbon. And we were also able to identify this by fish. So here we are showing three probe break apart probe, basically showing here like in interface like a non separated mecum locus, and here we have here like in this arrow showing it the separation, basically confirming this finding. So it's very encouraging seeing this is also OGM is also like being able to confirm these cases. The second case that I have is this case where we see relatively like a simple karyotype with a translocation between chromosome 2 and 3 and 5 and 6. And in OGM, as well as in the previous case, we were able to rescue, and we see those rearrangements as well. So he see again in the circus plot, the ribbon showing the translocation between 2 and 3 and 5 and 6. Having this zoom in into this mapping, we can see very clearly how this is looking when we have the mapping. So basically, we see here like a clear translocation between TATA and MECOM. This is a translocation, the TATA gene with MECOM that have been reported before. So again, like it is a confirmation that we can see this other, not as common as the classic inversion, but we are able to identify them. The next case, these are cases that where we are starting a little bit more of an impact of like what OGM is bringing to these cases. So this is a case where we see a karyotype that is not showing any involvement in chromosome 3. We do have some other alterations in chromosome 6 and 7, but chromosome 3 didn't show anything by karyotype. And in this case, when we look at the OGM, we were able to see that the chromosome 3 showed these rearrangement with chromosome 6. And this is a very interesting one because it was a relatively small rearrangement. Basically, it was an insertion of MECOM into chromosome 3. This is an inversion that is fairly small, so we want to be able to capture that by karyotype. And on reflex fish, we were able to see that again, like this 3 breakable probe showed that one of the probe was being located somewhere else. Again, like by fish or by karyotype, the resolution, we wouldn't be able to tell exactly what was happening exactly. So this is a case that basically, in this case, the diagnosis changed from NDS to AML. So very impactful in this case. The next case that we have is a different version of what we could have. Like this thing is a very complex karyotype. As you can see, we have multiple chromosomal losses. We have also additional material that we're seeing. So very complex. But again, like difficult to see what's happening in this case in more detail. When we look at the OGM circle plots, you can see a lot of the findings that were done by karyotype. We're also seeing here, you can see many of the losses here shown like in these orange areas. And we also see a couple of rearrangement with chromosome 3, and these are basically within chromosome 3 we have one, and with chromosome 3 and 6 we have this other one jumping here. This is better shown here in the diagram to the right, and basically it's indicating that MECOM is involved in 2 translocations. So again, these are a complex case, but clarifies the importance of having this resolution by looking at the MECOM rearrangements. These are not well characterized partners in the literature. They are not clearly reported. So the mechanism on this one is slightly not, is like not well understood as the one with the classic GATA2 and MECOM rearrangement, but it's still important for the stratification of this case. And another case that we found was this case that on karyotype looks very normal. As you can see, they're like not really anything happening in the karyotype. However, we were able to see an inversion in chromosome 3. This is not a classical inversion. This is an inversion that is involving different subsets. So again, smaller than what we have seen in the typical Yara mecom. But as you can see from this OGM map, we see that we have two maps showing clearly that they are like at these locations, this region between uh, 160 and 169. It is mapping and is being supported by the two different maps here, basically showing that it's like a clear inversion of this. And these are uncharacterized partners, but it's still involving mecom, which is still like under the WHO um, clearly a definition for like how this is a case that we have like a very drastic change from a normal karyotype. And the last case that I have, it's a case that we have a failed karyotype. The karyotype was not able to be completed. We didn't have enough metaphases to really have like a reading into what's happening here. So having OGM as an alternative option to try to evaluate this case was very useful. And in this case, what we see is that OGM reveal a mostly normal genome with translocation between chromosome 3 and 8. And in this case, it involved the MYC loci. And as you can see, here's a very clear translocation. So basically, we are here showing that this area on MYC, which is known to contain a lot of enhancers, it is translocated to MYC. And this is previously reported translocation, and this specific location in the downstream location of MYC is known to be involved in AMLs as well. And the mechanism here would follow the same idea that you have hijacking of a promoter or enhancer region that will drive the expression of MECOM and dysregulate cellular processes. So this is basically also a case where we can see the potential of how OGM is giving us a lot of information that we can use diagnostically. So in conclusion, basically kind of like highlight that I want to bring is that the cohort that we have evaluated identified 9 cases with MECOM out of 147. This is a 6.1 percentage. And the yield here was pretty high. We have like among those 9 cases, 4 of them were show cases, so show changes in risk and stratification. So basically this shows how important is this tool in our practices for these cases. And overall, like this basically also highlighting that we can also confirm other findings that traditional karyotype and fish are able to detect. Uh, Thank you.
Thank you, Nicholas. That was great. Can you remind me what you said that in 4 cases it led to changes in risk and diagnosis. Was it the same number that I just wanted to know what number of cases MECOM wasn't actually detected by the conventional approach? Is it
So it would be these 4 cases here.
okay?
So either in this case, we didn't see necessarily they were either cryptic in chromosome 3 or show normal karyotype, or we didn't have a karyotype. So these 4 cases involve MECOM, and we wouldn't be able to know that by a karyotype or fish. Well, fish,
Yeah.
yeah, but only if it's done upfront in
And,
some
and
of
so
these.
this is often the problem with most of our fish panels is that we sort of go into them. So if you have an upfront fish panel that has MECOM on it, you would catch some of these, not all of them, because we have a case. I think I can't remember if I discussed it with you before, Eric, or not, but we have a cryptic MECOM insertion. When I say we, I mean my data from UHN. Um, we had a cryptic insertion of MECOM into chromosome 3. It was about a 1.5 megabase insertion of material from chromosome 12 and 7. And invisible by karyotype in the MECOM. It wasn't even big enough to displace the MECOM probe. So completely invisible by conventional approach. So I think this is one example, a really nice example of a disease set that's really important to detect, but I'm sure it's not the only one. I think Newt 98 and some of the other rearrangements that we're now seeing with higher frequency because they're just not we just haven't been able to appreciate them with conventional approaches before.
Yeah, absolutely.
Any other questions for Nicholas in the chat? Raise your hand.
And Nicholas, thanks for that overview. That's a great talk. And as you know, since we've uh, put this together, there have been at least 3 more kind of unsettling ones. So yeah, I think we don't do fish from ecom up front, and that's probably again why this is that added yield. If we did fish from ecom up front, we'd find most of these, but it's kind of the same with any question, right? Like if you fish for everything, you'll find everything. The unsettling part is a lot of these are presenting as like MDS excess blasts, and then obviously they're behaving badly. So it's likely, in the years preceding this, we're missing a lot of MECOM rearrangements that we just assumed were, oh yeah, MDS monosomy 7, they behave badly. Just happens to happen sometimes, right? But
Yeah.
the number of these that are showing up as MECOM rearranged, and in retrospect are not surprising because they have pathology that totally fits. It's unsettling, and people in the BMT ward are noticing.
Yeah, fantastic. Okay, if no other questions, I think we'll move on to the next case. I would like to invite Flesher to join us.
Hi.
Hello, Flesher.
There's a comment in the chat as well.
So here's a question in the chat. I'll just read it out, and then Nicholas and Eric, maybe you guys can take a stab at it. Sorry, Flesher, 1 minute. Well, we can't. So we can't fish everyone. So is there a population you would recommend that we fish MECOM? Anything greater than sort of suggestion? Anything greater than 5% blasts? So I'll throw that.
Eric, I let this one for you.
I don't have a great answer for you, unfortunately. Before we had OGM, honestly, I found a good number of MECOM rearrangements just kind of being in the position of seeing the karyotype and also knowing the pathology of it. So if you have pathology that's suggestive and you have that monosomy 7 show up, I kind of go, oh, I don't like that. And, I've reflexively thrown MECOM fish when it doesn't look at all MECOM rearranged because the pathology fits. And if you have that, terrible dysplasia, especially megakaryocytes, excess blasts, and platelets are preserved, like that to me is suspicious. And we used to reflex a lot more MECOM on that basis, but now we kind of know, well, we have OGM. So, a better test is going to solve a lot of those problems. We do have hematopathology-based triage a lot of the time. Yeah, so for like fish, we tend to do that. The karyotype, if it gets signed out before the pathology, which is not common at VGH, it may just go missed as a potential thing. But even then, that's kind of a nuance that if no one on either side is recognizing it, then you have to have a test that'll look for it, whether it's fish or OGM or something else, or maybe you're just missing it.
Yeah. I mean, at UHN, we used to do the any 3 that looks funny gets fished for MECOM, which is probably a good rule, but it's not 100% either, because as Nicholas showed and as our experience has shown, you can have totally normal looking 3s, cryptic insertions and other mechanisms that don't displace enough material to not see the translocation. So, okay, fantastic. Thanks for the question. Oh, Hubert, that was you. You're being quiet and just putting your comments in the chat today. So anyways, on to Flesher. Flesher, the floor is yours.
Yes, thank you. We were having, in fact, the same approach here for MECOM, but yeah, it does help us to understand why so many of our intermediate AML were not behaving correctly as it used to. So yeah, we came through this case a few weeks ago of a woman was already known to us. So she was in relapse, and we happened to know that she was in fact being engrafted. So the woman was diagnosed with a monocytic AML in 2023. The karyotype was a failure at this moment. We did the RECGH, and we were able to detect a trisomy 1q, a deletion 7q, and an homozygosity of chromosome 21, which in fact was reflected by RUNX1 mutation in 93% of the cells. So this patient was allografted in early 2024. And she came back with a relapse in October 2025, which at this time we were able to perform OGM as a first-line assay. We didn't know what was the sex of the cord blood, but it happened really fast to us that a sex mismatch was detected with VIA. So we were detecting a male. So we assumed that the graft was a male. And in fact, this kind of mismatch was observed also when we did our in silico dilution assay. And this mismatch would happen at 15% of sex difference in the two samples. So how does it look like when you do the OGM? So you can see that there's a lot of imbalances, allelic imbalances throughout the genome. We can see still the trisomy 1q, the deletion 7q. I will come back again with the new anomalies, which are the deletion on the trisomy 2q and the deletion 8q. We could see the mismatch with the sex chromosomes. So in fact, the gain on X gave us an estimated copy number of 1.186 with a loss of Y chromosome, which is almost the same, which was compatible with the chimerism with about 78% of the cells from the donor and 20% from the recipient, which was in the relapse. We did saw the anomalies that were present at the diagnosis, and also two anomalies that were confirmed by FISH. So we saw a deletion of MYC in 20% and and ALK trisomy not in 80%, pardon me, it was in 40%. So again, you can see those allelic imbalances. Later on, the chimerism was confirmed also with the molecular assay, but we were happy to be able to confirm it to the medical team. So that's how I decided to run the final ISCN. In our center, we don't like to use the VAF for the derivative chromosomes. We prefer to use the estimate copy number everywhere, so it's less confusing. So that's how we decided to present it. It was quite nice.
Great. Thank you, Flecher. Any questions for Flecher? It was really nice to see that case, Flecher, and the allelic imbalances in VIA, which of course we've run some of these post-transplant cases in OGM and just analyze them in Access prior to the guided assembly, where we didn't have any allelic imbalance information at all. So
yeah,
just, you could see the sort of distortion of X and Y, and that was about it. That was all you could get. So
yeah,
that's kind of really interesting to see that, and a nice group. That's really good. Sorry, any other questions for Flecher?
Great case. I think we had a similar one recently that was a sex mismatch in the other direction, and writing the ICN was an interesting question for the group, as to, especially with extra X chromosomes and an XY, like what's the proportion?
yeah.
I think we ended up using FISH to actually resolve that question, but I like the way you wrote the ICN, because I don't think they give you guidance in the book about chimerism with OGM.
No, not really.
It's not there yet. I think it's a good point. But I think it's tricky because I don't think it's straightforward. I don't know that there's a really great answer or a really great way to do it. And it's not just an OGM problem. It's a problem across all technologies that do any kind of bulk genome analysis, right? That's not a full single cell representation of a chromosome. So yeah, I don't know if there's a really great. We had a paper that came out in Molecular Cytogenetics that called "What the VAF." I don't know if some people have seen that paper or not, but it gives some guidance. I mean, it's a paper. We were already, I think, 3 or 4,000 words over the limit when we submitted it. So I think, it's one of those things where there's just so much to talk about in these areas, so many intricate, nuances to the problems. And so you actually reported in the square brackets, Felicia, estimated copy number and not proportion of the sample, right? Okay.
Yes. Well, no, excuse me. It was the proportion of the sample. Yeah.
Proportion of the sample, yeah.
Yeah.
So
and not the VAF for the structural. So having a mix, it's quite confusing.
yeah, I saw that. So when we originally wrote the OGM nomenclature paper, we did not include VAF as an option in reporting OGM structural variant frequency. And I noted that in the actual ISC 2024, that is now given as an option, although you have to be clear that you're reporting VAF and not proportion of the sample. Proportion of the sample is consistent across the rest of the ISCN, which is why I think why we use it. But it is not entirely straightforward, when you have these situations. Sometimes it's just there's so many underlying assumptions about what the composition is of the abnormality that it's difficult to actually define a clear proportion. So anyways, that's something for us to put on the agenda as we get more data to try to figure out how to do this more, see what things fall out and from the technology, if we can figure out a better way to do this. But anyways, thank you so much for presenting, Felicia. I really appreciate it. Okay. Any other, I think that's it for the presentations. Eric, you have one, I believe, right? You have a case?
Yeah, I have a quick case I can show you guys because I saw it and I was very excited having seen it, and I'm sure some people appreciate it.
Yes.
And then after that, I can just go right into the survey if you'd like.
Perfect. Sounds
Great. So this is a case that came in a couple weeks ago or a month ago now. Woman in her 50s who had received chemotherapy for breast cancer a couple years back and presented with pancytopenia and occasional circulating blasts. Obviously had a bone marrow done, and the findings in her bone marrow included more than half the cells being blasts, which were fairly big, basophilic, have this kind of wispy or elongated kind of pseudopod-like cytoplasm. And some of the blasts were doing this very unusual thing, eating red cells, so erythrophagocytosis, which you don't see very often in AML. These were monoblasts per flow cytometry, and bone marrow was largely effaced. Had a karyotype done, which we always get before OGM, obviously, it takes less time, and showed a translocation 815. And for those familiar with AML, with the erythrophagocytosis in the story, it's kind of expecting 816. So I was a little disappointed to see this, but thought maybe it's some unusual variant of an 816. But because of the breakpoint and the potential for change the diagnosis quickly, looking at BMT wanted us to look for FGFR1 rearrangements. So we followed this up with FISH. Sorry, just another karyotype showing the same thing. And as kind of expected with the phenotype, you see the interface. I mean, we have an 8 centromere control here. It shows clearly that the FGFR1 locus has moved away from the 8 centromere, but hasn't split. So it's not FGFR1 rearranged. This is a metaphase showing the same thing, not great quality. OGM got interesting in that it showed this translocation 815 that we knew was there from the karyotype, but also that this was a 3-way translocation involving 16. So because the breakpoints on both 15 and 16 are quite distal and pale, I don't think we'd expect to see any difference really in the morphology of the 8 that results from these. But we do see the big chunk of 8 moved to the 15 rather than the 16 in this case. And it's a little bit of a closer view showing this 3-way translocation loop. And as expected from the pathology, this is actually a Cat68 CREB-BP fusion on the 816 rearrangement component. So this, oh, sorry, I had them right there, didn't I? This translocation 815 is actually a translocation involving 816 and 15 that has the classic morphologic findings. So when you see this reported, erythrophagocytosis, giant blue monoblasts, that's kind of where you think maybe Cat68 CREB-BP is the culprit. And in this case, it was a bit of a hidden culprit. So I was excited to see that because that's my first one I've ever seen in real life. I think I've seen one in an archive, but they're quite rare. Any questions about that before we move on to the survey? All right. So survey, acknowledging that I sent out this survey relatively late. Thank you to those who responded, and thinking perhaps in the new year I'll check in with BioNano and see if they're able to give me a list of, consenting people in Canada who would be willing to feedback more comprehensively on who's actually using OGM in Canada, whether it's research-based or clinically based, and get a better idea that we can really map that landscape. But for now, the responses we have will work, and I think it represents most people who are using it clinically that I'm aware of. So starting with a question, who are you? Where are you from? We have the two sites from BC using this test. They have North York, then scattered throughout the east, and University of Calgary responded as well. So representation, a lot of the country. People who've responded in the past, I think, so thank you again. Where are we with OGM implementation? So most people who responded are using the test clinically, at least in one context. And the remainder are split between here to learn about OGM and in the process of trying to get an instrument, which I know for some people can take some time, took quite a while for us. There's a smattering of people also responding that they do send samples off-site, but analyze on-site, and that some people have the platform on-site but are in the context of validating, I believe. Either is not shown. Oh, perhaps there's not shown. Yeah, I think there are no responses for those. Sorry. OGM instruments people have are pretty fair split between having Sapphire and having Sapphire and Stratus. And then, of course, about a third have none. I think as expected from what people are doing in the literature, most people who are using it for hematologic malignancies and primarily myeloid is the most common indication, but some group using it for myeloma, which we know, and we'd love to learn how you're doing that and how it's working for you. What indications are you considering? I think this reflects what people responded when we last sent this out a year ago. Again, most people that want to implement it are looking at hematologic malignancies, whether it's beginning to use it or expanding it. One response, solid tumors. I suspect that's our lab. And then the constitutional testing. I didn't look at who the responses for those are. I know that's been looked at in some labs, but it would be interesting to see that roll out. And when people begin to use it for that indication, it would be great to have you discuss your experience at the community of practice. This was a question that was requested from someone in response to the meeting invite. How many specimens are you putting through per week? Pretty standard across the board. It seems around 6 on average is what people are seeing go through. I know it varies quite a lot depending on volumes in the lab, and ours are generally in that range, but obviously weeks where we have a whole lot of specimens and we have capacity on more than one instrument, it goes up. But it seems like around 6 on one Sapphire is pretty standard. And EQA is something we discussed at length in our last meeting. So the question came up, how are people doing this? And right now, most people who are doing it are using interlab exchange, which is also what we're doing. Commercial EQA, I'd be curious to know who is using that and how that's going, because we haven't yet explored that, but that's something obviously of interest. And then blinded reanalysis in one group. Pipelines currently in use. Again, pretty even split between rare variant analysis and guided assembly. We personally have been using primarily guided assembly since it was validated here. So haven't really gone back to rare variant or de novo unless we suspect something's gone terribly wrong in the guided, but I do recognize the differences are not always good. And then de novo presumably is in the context of constitutional, but I'm not sure. Does your lab use a tiered response? Yeah, everyone's using a tiered reporting framework for OGM. So this would be interesting to know, I think, in a future discussion, what people are doing, how things are being reported, because I think it's largely going to be similar, but probably nuances very different. And this kind of fed into the last point of discussion for the agenda is what are your lab's pressing concerns with OGM? And I think most people would expect the responses that came up, things like report automation and how to scale up testing, especially with the current instruments available. Some responses included concerns with the reagents, chips, and instrument quality, which in this response are noted to result in recurrent failures or delays in turnaround time, probably because of failures. Technical support not elaborated on. I'm not sure if this refers to the obviously change in support architecture with the company restructuring. Obviously, the things that we all know are problems with OGM, things like limitations of ploidy detection, small clones that are maybe below the limit of detection, which we see a lot in, MDS now that we're testing for that, and how to report things properly when you find things that are unexpected. The viability of Stratus, which I think is a topic of discussion in general here, automated DNA extraction, and how to improve turnaround times. So again, small number of responses, but I think reflects a lot of what people are talking about at meetings as well. I see there's a response in chat here. Would you distribute the survey results? Absolutely. I will send that out. I can send this out right after the meeting, and I'll see if there's any further specifics I can pull out of it from what's in there, but without identifying who responded to these. And then, like I said, if people who are using BioNano might be okay with it, I'll ask our representative at BioNano if they could perhaps contact Canadian customers and see if we can more thoroughly contact people who might be willing to respond to what are you using this for, what are your problems, and where are we going with this? Because I think obviously sending these responses out, these surveys out is capturing a lot of people, but I think BioNano is in the best place to know who has these, who's using these, and who we could best consolidate information from. Are there any points of discussion from the survey or questions about that? I think it's kind of what we talked about a year ago and hasn't changed a whole lot, really.
And I think some of these points are also in the open discussion as well. So we can do that. But for Flesher, you have your hand up. So
Yeah. Last year, Eric, you were talking about the problems with the strategies that you were having in the house. We did face the actual same problems during the spring, so that in June we decided to not run anything else on the Stratus. We haven't decided yet to decommission. Well, it's decommissioned, but it's not returned to the company. We are still showing some opening to BioNano. So to help them to see if it's possible to have the Stratus being working correctly. I would admit I am a bit not confident about it. But well, at least it gave us a way to be able to pursue our Sapphire rental and paying our reagents the same price that what we were having, so at $450 per genome, instead of the increase at $700 per genome with an instrument rental. I hope that it would get fixed. They say it's informatics, but I'm not really sure that it's the case. But I will keep the group posted. So if at some point it works correctly, I will give news. But if it doesn't, we will probably consider buying the Sapphire that we are having now, which will be in fact 3 years old in the spring. So buying it at a lower price and probably getting a second one for our lab too.
Flesher, what exactly is the issue with the Stratus that you're having?
We were able to see the images of the molecules that are captured. Brett came during the summer and he was really open with this. So he showed me the images. So we could see that the molecules are really fade. It looks like there is no DAPI, in it, but we know that it's the case. So we have run the same sample on the sapphire and the stratus. So we were having both images, and it's really, really fade for the blue on the stratus. So since it doesn't see the backbone of the DNA, it doesn't count the molecules. So yeah, that's what
Because we're not having
the
that
problem.
problem with our
Mm.
stratus.
Okay.
So
And what do you know? What is your number?
I don't.
Serial? Because we
I,
were having,
I don't know.
I think, the
Michael's
10th
on
instrument.
Michael,
So was it in the first ones or the second batch?
no. We have a much later one. Yeah.
Yeah, maybe they have fixed it
Yeah.
I know I've
without
heard of
knowing
laser-
it. Yeah.
I have
That's
heard of
what.
laser problems, and I think our issues have largely been mechanical, at least the ones I'm more familiar with, like things getting stuck inside the stratus, things not wanting to move from the carousel to the imaging stage, failures in contact with the, electrical components that move the DNA through. So it's kind of been a whole bunch of different things that have been going wrong that I'm aware of, but I'm sure there's more that I'm only peripherally aware of. So I see we have a couple of people with their hands up. I think Hubert had his raised first.
Oh, thanks. This was on a different topic, but thank you for doing that survey and showing some of the responses. Do we have anything to say about the benefits of OGM, specifically from the end users, say the clinicians, that might also be something very useful to provide BioNano and say, hey, you know what, this is the current state. This is where you're filling a particular niche. And so like get your act together, or
I
think
haven't
some of the
solicited any surveys from people, but I'm sure the Canadian Leukemia Study Group could help with that kind of thing. I know our local BMT group has sent accolades in the past saying this is better, like this changes practice. So we can easily pass that along. I think we have, but I think it's clear, at least from our group, the clinical groups are very, very happy with the testing.
Yeah, great. So I'll touch base with you after, maybe trying to assemble that piece, which is also very pertinent, aside from the struggles within lab, to show what clinicians are, very happy with, including if, exactly which indications perhaps we really want to focus on here, provide, again, practice-changing care right now for patients. So thanks again.
Thanks, Hubert. Tara, you had your hand up as well.
Sure. A couple of comments. So I guess just tailing on what Hubert was mentioning, we have done a very nice clinical utility study in our lab that's currently under review. Hopefully will be published in the new year, awaiting acceptance. And we did our clinical utility review of the first 200 acute leukemias. And the evidence is strong that it is showing a very nice clinical yield, both in terms of restratification for risk as well as change in diagnosis. So Nicholas provided a nice small cohort, but these are not rare instances. We have many, NUP98 rearrangements, as Adam mentioned. I think we had another one last week that was not apparent at all, obviously, by karyotype. As well as KMT2A, MLT10 rearrangements, and quite a few others. So we think it's really quite impactful. But it is tedious. There are challenges, absolutely. And I think that that's one of the benefits of a group like this that can transparently discuss some of these issues that we're experiencing. Tailing on the stratus discussion, we also have a stratus and a sapphire in our lab, and our stratus has just been chronically failing for so many issues that we can't even place a name on them. So we've decided to decommission that instrument and return it to the vendor. They are providing us with a sapphire using a reagent rental agreement as well, so effectively extending our sapphire agreement. So because we have to go through that procurement process, it is quite slow, but we are hoping to get a replacement sapphire in the lab, and BioNano actually has been quite supportive of that. So I would suggest don't hesitate to contact them and try to negotiate that for yourself, Flesher. Yeah.
And
Thanks,
it really does depend on the lab and your volume situation. I mean, even at UHN, we did capacity calculations. We could adequately address, over 1,500 samples a year with 2 sapphires. So depending on where it is you want to scale up to, You know, and it really is going to depend on the volume of leukemia. And then I think for labs that don't see just leukemia, I know like Tara and Eric and the UHN lab are in a sort of specific niche where it's very leukemia focused. Certainly other labs that have a broader perspective, perhaps, that want to move into constitutional, you know, because there's issues around testing with repeat expansions, for example, that you know, could be expanded, as well as just general constitutional testing. You could potentially see volumes exceed the sort of, you know, reasonable capacity of having a couple of sapphires, where the Stratus would maybe make more sense.
I think that's a
Okay.
good point, Adam, just to kind of tail on that as well. You know, we're also running for chronics, and it allows us to give a little bit of wiggle room for those scenarios where, you know, we would like to we maybe have a lull in our acutes, then we can run more chronics. We're definitely struggling with one instrument. It's a challenge for sure, and we need to absolutely to maintain those turnaround times. I think we'd also like to expand to CLL as well, and we're validated for that, and it looks nice. So yeah, I think that there's definitely, you know, a need for one reliable instrument minimally, but probably a lot of labs that would require two.
We are very happy having the two instruments, and I think that would lend itself to a lot of success at Kingston Health Sciences Center. The fact that we had a production instrument and a sandbox instrument allowed us to pivot between the two instruments, and we didn't jump onto the Stratus story. We're just waiting to see how all that played out, and I think that has served things well for the center. But yeah, I would agree with Tara's statement, having the ability to have a backup instrument does help a lot with flexibility. And Naseem, also, you have a question, so.
Yes. Actually now in Hamilton Health Science, it's been a while that, you know, they are using OGM in the research. So the main part first started as constitutional testing to replace CMA, and at least 100 cases were, you know, sequenced. And we started, you know, to look at the data, and half of the data is analyzed until now. And basically, we are not happy with the data that we got from the OGM. And they had lots of issues with the instrument. We have one, you know, Stratus, lots of issue with the chip. So we got new chips because, you know, the chips were not good. They replaced it. But the issue is most of chips right now are, you know, they are expired. And because of the data and everything, we decided to stop the constitutional one because, you know, we don't really get anything, and we cannot basically replace CMA. And then, so because we had lots of chips, we decided to basically pivot to CLL. And these chips is for blood, peripheral blood. We could not use for bone marrow, so we started to use for CLL. It's a new, you know, a new project, I would say. We ran several CLL cases, and until now we analyzed a few of them. And actually, based on what I saw in the few cases, it seems promising. So we offer only the FISH panel for CLL, but in CLL cases in which we did, you know, OGM, we got lots of stuff. So I think, you know, for CLL, probably it's worth to try. And probably in near future, we can also share our data to the community.
Yeah, I'll make a quick plug there. As we do really appreciate, like, if you have any suggestions, you have a presentation, if you have some pilot data and you wanna segment at a community of, we will have more meetings. We will have them more regularly, and if people have stuff to present. I mean, this is, you know, I mean, God bless Eric McGinnis, because you keep reminding me to schedule meetings, and we keep doing them. And I think it's really great. But if there's more data out there and you're willing to present, please let us know, because this is what helps the community, is everybody taking their data and contributing. So I'm going to shut up now and let Flashare talk.
Yeah, I did write in the chat about comments that I received from the clinicians regarding use of OGM. What we do in the lab is that we will freeze cells from all bone marrows that enter. So whatever the indication, so we do have surprises. So a doctor that thinks that this patient is having an MDS, and finally there's a full lymphocytosis. The flux, the cytometry is ambiguous. The pathology says, well, I don't know, maybe it could be a marginal zone lymphoma, but maybe it's a DLBCL and things like that. And if there's enough lymphocytosis, something like 30% or more, we will do OGM. So, and we are able to tell them, well, we see that there is no translocation, so you can exclude the DLBCL with make BCL2 and BCL6. We see a trisomy 7q and a trisomy 3q, so it goes a bit more with a marginal zone lymphoma and things like that. So it's that kind of unexpected use of OGM that we are starting to do. And the last case I'm about to have the data is a patient that the clinician was sure the patient was having an MDS. And finally, there is 25% of multiple myeloma plasmacytes in it. And I told her, well, before doing another marrow to your patient, I can still take a look and see if we see some rearrangements. I will tell you what are my limitations, but I know that at least for the translocations, we are able to see them at 5%. So it could be possible that we can issue an interesting result for you. So that's what we are being told by our clinicians recently.
Thank you for that, Fletcher.
Thank you. So no one is having issues with the sapphires, right? Or everybody's happy with their sapphires? I just want to, so we went live with all of heme in March 2011. We were waiting for the software upgrade, which now turned into a hardware upgrade because we need the top hat functionality for everything. It's been fascinating. Every time I sign out a case of, CLL or whatever, we found cryptic FGFR translocation, or actually it's an insertion, but it was very small to be seen. It's been like signing out the cases has been a lot of fun, but we have this up and down in service. Like we have 3 months of very smooth, fantastic runs, and then 3 months where everything goes wrong and you don't really know what's happening. There's no way to troubleshoot anything. Like the whole thing just falls apart. And honestly, like even if you take things step by step and start to troubleshoot, it's just so finicky and nothing works. And then suddenly everything starts working again. And I haven't learned anything from the issues we've had before. I personally think they are the chips that were wrong, but BioNano insists they are not the chips. We blame the instrument, we blame the electrodes, we blame the the technologist, we blame but not the chips anyway. But we still think it's the chips because suddenly everything started working the same, and we didn't change anything because we didn't know what to change. In terms of multiple myeloma, I'm having fantastic results using the Milteny columns and for separation, so isolation of plasma cells. And I'm hoping to go live soon. I just need time to analyze the data. And again, those have been very, very nice. We are comparing our samples to those that we sent to Mayo and UHN for also isolation for multiple myeloma. In terms of constitutional genetics, my experience has been we were able to reproduce, although we have relatively few cases, all the microarray results. Actually, we were able to, one of the cases, was missed by microarray and was detected by whole genome sequencing, and OGM detected that. What I'm having issues with is whole arm translocations. These are consistently being missed, not just the Robertsonians, whole arm, other chromosome translocations are being missed. And mosaicism, of course, that's obvious, right? Mosaicism in XXO, XO, so those don't work. For me, what I've done at this point is for paternal and maternal miscarriage or infertility screening, I'm sending those to karyotyping first. Because these seem to be the more frequent ones, I'm thinking about doing both tests at the same time. But in terms of microarray, again, I have very small cases, but we were able to reproduce each and every case of microarray that was run by OGM on the Sapphire, as well as each and every case of whole genome for structural variants. I was reading the notes in the comments. Thank you. That has been our experience. They do respond. The response rate for us, the response rate from Bionano is relatively okay. Not as good as my other, like Thermo or Illumina, but they do respond. And the troubleshooting is always the same steps for us. We just repeat the whole SOP over and over again. And we just, spent a couple of hours of the entire lab staff sitting there trying to go through the SOP, which is something we are doing. And then we personally think it's the chips, but they don't. And we thought it was that's where we are. They are responsive, they are cooperative, but there's a disconnect somehow. Yeah. But the signing up the cases is fascinating to me. So thank you for I'll just stop talking now. Thank you for letting me talk.
Thanks, Doha. I think there's lots of lots of stuff to talk about. It sounds like Tara, you had your hand up there for a minute. I don't know if you still have a comment.
Sure. I was just going to add to Doha's experience that typically with the Sapphire specifically, we don't have chip-related issues often. Occasionally, we do receive an error when we put the chip in, but then basically just trying it again seems to work, and we don't have to exhaust that chip to do so. But we do have a lot of issues with reagents, as I see Eric also and Adam mentioned in the chat. These can arise from many different things. For sure, there's some QC issues on Bionano's end. To troubleshoot those, we basically just rerun a sample on a different lot, and then we show Bionano that it currently is failing using a given lot, which is honestly a lot of work. But then we request replacement of all of those reagents. So once we demonstrate to them that there is a recurrent reagent issue with a particular lot, they do tend to replace them and often comp those specimens that failed on a poor lot. We've also seen this with, we often, at least in our lab's experience, have challenges with shipments across the border, and occasionally reagents do get stuck at the border. If they arrive and they're thawed in any way, then they almost inevitably fail. So I would just maybe mention that as well. If there's any delays in shipment, then that's something to keep a close eye on, because in the past, we have had issues with that, which seem to mostly be resolved now. And I would also just echo that Bionano is most definitely not helpful. They always indicate that it's something wrong with our lab, something that our technologists are doing wrong. And we do typically have to combat that with, proving that it isn't our technologists, that it is really related to given reagents.
Thank you, Tara, for that feedback. That was really useful points. So I see also Fleche was talking about refusal of the thawed reagents as well. So
So the question too becomes: are the reagents being shipped with like temperature indicator strips or anything that would, indicate that they have sat at room temperature for a long time? And then that would be good. That would be good to just not accept them. The other thing too is that one of the things we could maybe do as a group is that if somebody is having problems with a particular lot, we're probably all getting the same lots to some extent. So if you are having problems with a lot, maybe we should figure out some kind of system that we can just sort of pass that around. I don't know if we need to have like a list server. We could maybe we'll have to we can figure that out later. It's a technical problem, but anyways, Eric, go ahead. I think that's a good suggestion nonetheless.
Cynthia
I'll
Chow.
remind everyone we do have a listserv that's never been active. So I'll send that out again after this meeting. I know there's some people on it, but it's never had any activity. So I
Yeah.
can send them back around, and everyone can flag that as do you have a bound and a problem? Send it to the group and see if we all share the same problem.
Yeah.
I think
Cynthia
that'd be
Chow
great.
also mentioned in the chat that she's noticed that there's a precipitate often which dissolves out with vortexing.
But is the precipitate, Cynthia, is the precipitate
In
indicative
the
that
labeling buffalo Crocker. Sorry.
No, I know, but is that indicative that there's a problem and that even if it goes back into solution, that the reagents still don't work?
No, it will work after. We have the same.
Okay, well, good to know.
Small crystals.
Yeah. Okay. Let me jump on to I just want to because I feel like we could probably commiserate on reagents and Stratus issues for a while still. So let me just touch this really briefly. We wanted to touch briefly on the hypereosinophilia cases, which I think is sort of an interesting non-technical problem. I think it's a biological problem. So I'm actually really happy to get a chance to discuss this with everyone. So let me just pull up my presentation here really quickly. And then hopefully we can get an idea as well by discussing with everyone if everybody's sort of experiencing this sort of similar problem. I think I've certainly discussed it with more than a few people. So I think it's a widespread problem. I would be curious to hear if anybody has any different experiences, and if you know what the magic sauce is to make the problem go away, we'll be I think everyone would be very interested to know. One of the assays that when I was at UHN we launched, one of the first assays we launched was testing for eosinophilia or hypereosinophilic syndrome. And so obviously, according to the WHO, there's a series of translocations that are associated with these. If it's related to a neoplasm, of course, there's a lot of other there's a lot of other physiological conditions where eosinophilia can occur, specifically in response to infections, whether they be parasitic or fungal bacterial. You can see this in allergic disorders and skin diseases, graft versus host reaction, and all sorts of other stuff. So eosinophilia in and of itself, and I think probably most of us will know that we'll test 100 eosinophilic indications before we'll actually get one that we'll find a genetic cause for, where we find a rearrangement. They tend to be the rearrangements themselves: PDGFR alpha, beta, FGFR1, ETV6, ABL1, FLT3 rearrangements, and missing one, JAK2. These ones that are in the WHO as the MLN, the myeloid lymphoid neoplasm with tyrosine kinase rearrangements, specifically that result in eosinophilia, are the ones that we're obviously looking to detect, and where we think we thought OGM was a really excellent platform for doing this. One of the things I learned very recently about the biology of eosinophils that I found fascinating was the fact that they are associated with what they call these EETs, these eosinophil DNA extracellular trap release functions, which is basically there's two kinds. There's the genomic EETs, which is where nuclear DNA is released. So the membrane, both the nuclear membrane and the cellular membrane, open up, and these strands of DNA actually extend out into the not the cytoplasm, the extracellular environment. And this is not only a stimulus to the immune system itself, but also it serves as a trap for foreign materials such as bacteria or helminths like worms and other types of parasitic infections. So what you end up with is a situation where you have this where you have all of this material that the body is fighting and releasing all of the proteins that are in the eosinophils in order to help degrade, digest, and destroy them. And of course, this is all very, I think one of the things here is, of course, that by extruding this material outside the cell itself, it does protect the cell somewhat from damage and focuses the damage on the thing that the immune system is trying to fight. So you have the genomic EET extrusion. You also have the mitochondrial EET extrusion, where the DNA is extruded from the mitochondria instead of from the cellular nucleus. We have these two types. And I say that a total aside, just basically to suggest that, the eosinophils are filled with really highly toxic proteins, the major one being the major basic protein. And the major basic protein is highly toxic to parasites, bacteria, and mammalian cells by disrupting lipid bilayers. So I think where I'm going here is that for those of us that have tried to run these assays on OGM and noticed that we've had problems with them, and we think probably it could be related to the fact that the contents, when you get eosinophilia and you have many more eosinophils in a sample, and then we basically blow these cells up to try to get the DNA out of them, we're also blowing up all the stuff that's in the cells and releasing stuff like major basic protein and the other proteins that are in there. So where are we with this situation? We have a problem where we often get samples with good cell yields, but terrible DNA yields. One of the sort of analogous pieces of evidence we get from fish assays is that when we do fish for eosinophilia cases, they often have very high background. And if there's anybody on from the UHN Fish Lab, maybe they could comment on that. And then cause I kind of got there already with the possible release of MBP1 and other proteins that could be basically just rapidly digesting DNA before it can be cleaned up. And then is there a solution? I think we've sort of discussed like how could we maybe do this? One of the things that I read is that heparin is acidic, so it's an inactivator of MBP1. And so, like anecdotally, do we have any evidence, for example, that sodium heparin as an anticoagulant collection tube is maybe better than EDTA in these cases? Because again, I don't know if because the problem is that in the vacutainer, the buffering capacity is not really an issue because the cell membranes are all intact at this point. And so really, I think the problem becomes at the point that we begin to lyse the cells, that's where the potential for damage can occur downstream to the DNA. So anyways, that's my run up to the problem. If there are any experiences or comments about that, it would be really interesting to further discuss it. Eric.
Yeah, so this came up when we started doing this for eosinophilia. Again, hoping it was a better test. And I can share our limited experience because, as you know, these are not common requests, and we do screen them to generally make sure they seem kind of appropriate. These are the ones that we've attempted OGM for since the assay has been deployed for that indication. And the ones that have failed are highlighted here. And you can see the relationship between eosinophil count and failure, very, very high. All these ones here that worked were normal by OGM, had a pretty mild eosinophilia. So like this one's maybe the only one with maybe a good solid eosinophilia that fits with that myeloid lymphoid neoplasm eosinophilia, but the rest of them are pretty middling at best, or not even really eosinophilia. Some of them were done for other indications, like obviously neutrophilia that you can't explain and isn't positive for any of the typical rearrangements, but with some eosinophilia. But the ones that don't work are often the ones that have an eosinophil count that's quite high. So in the bone marrow, looking at the fraction of eosinophils like 50%, 60%, 30%, versus the ones that do work often have more, in the normal-ish or slightly elevated range. So 7%, 4%. This one, the 16% eosinophils worked. All these ones had poor DNA yield. The cells going in were great. Those are bone marrows that are very cellular. So the, pre-counts all look good, and then extract DNA and try and quantify it, and there's almost none left. This has been, like you said, discussed elsewhere, and at the International Collaborative, whatever, OGM group, it's been raised that a lot of labs are having this problem. And it's actually been looked at and published on by this group from Belgium and France, where they discussed this exact patient population. And in a small number of their cases, they did find that the DNA yield was quite poor. And they modified the extraction protocol to try and improve upon it. When you pull up the supplement, well, they describe basically adding PMSF to try and inhibit these proteases, presumably, and see if that improves the yield. And it sounds like for at least a couple of the cases, the yield did improve, but some of them still had poor quality DNA that's just not salvageable. So we're not sure the best approach to these yet. Most of the ones we've seen haven't been cases that we actually expect do have rearrangements, because they tend to be patients that have things like vasculitis or florid reactive state that explains the eosinophilia. But one did have a known FGFR1 rearrangement by another method. So that there are cases that we will miss by OGM because it just doesn't work. And obviously, we know the four common rearrangement partners that you might see work with fish, but if they're cryptic, you won't, or if they involve another partner, you won't. So finding a method that works and is not susceptible to this problem would be great. We're just not quite sure what that is yet. We spoke to Bionano about it, and our contact said we're not aware of this problem. We don't know what to do with that. I see he raised a question in the chat. Is it PDG for alpha translocation in just the eosinophils? Not usually. These are usually multilineage, so kind of like CML, it's in everything. The cases that, I mean, as you know, the myelolymphoneoplasms, eosinophilia often have eosinophilia, but also neutrophilia, monocytosis, other stuff. So it tends to be multilineage. And I suspect if we could get the DNA to actually function in OGM, it would work. Because if the tube is just full of goopy, damaged DNA, I don't think it's specific to the lineage that matters. I mean, whether eosinophils are destroying their own DNA or the DNA of the neutrophils that contain the rearrangement, I don't think it matters personally. But the fact that we can't get any DNA into the chip to work seems to be the big problem.
I'm actually curious about the using the isotachophoresis for extraction. Because that maintains everything in liquid phase from lysis right through to extraction, and it actually separates the components based on their electrophoretic mobility. So that may be one solution. And so I mean, clearly there's a technical issue here, and I think we've sort of been addressing it. We've been addressing it in a very an unstructured way, and I'm not sure if there's a really nice structured way to do it, because there's a lot of good questions here. Like Hubert's talking about shipment time. So like when you take the sample out of the body, is it basically a time bomb, depending on how many? And Eric, you've shown clearly that the eosinophil count makes a huge difference. If it's really eosinophilic, and then are these cells just popping open, and then it's just like a milkshake basically. So yeah, anyways, yeah, I think sort of dealing with some of these things in a structured way is going to maybe help us to do that. Tara, did you have something to add?
Just to really quickly add for Hubert's question, so at least the marrows, they're taken in-house, and we often freeze within 24 hours. So I think that somewhat minimizes that concern as much as possible, but we definitely haven't tried any alternative tube.
And are you usually using EDTA or heparin for your cytotech?
Usually EDTA. We accept both if necessary. Like if there's some issue where we don't get an EDTA tube, then we're validated for sodium hep as well. But almost all cases are EDTA.
So if people are doing this test or trying this test and have data about what has and hasn't worked, I think that'd be super interesting to see from a multi-site perspective what is the threshold where we need to worry about it. We know the ones where it's maybe not as indicated are going to work, but the ones where it's clearly indicated are the ones where it seems like the failure is very likely.
Yeah. Well, ideally, it would be great. It would be great to get a practical technical solution to this, whether it be a chemical one, a sort of a stabilization isolation protocol. I mean, I hate to suggest. I don't know. So, and I don't even know if there's something, in terms of a buffer, maybe a more acidic buffer to inactivate the activity of these proteins, or cell some kind of different cell isolation method.
This
And
group
then
recommends PMSF or something, so maybe I don't know if anyone's tried that, but I mean, we've
I
I
thought
think
we
the
tried.
problem
I
we
thought the
have is
whole point
that
was that we tried to
trying
get away from PMSF.
yeah, and the problem we've had with trying anything is that these are so rare that making modifications is really a one-off and challenging to evaluate systematically because these cases with real eosinophilic like this are quite uncommon, right? And if you see these indications, are mostly people with like an eosinophil kind of 1.2 that people can't explain. It's probably allergies or it's probably
But
vasculitis or something that's just
but
reactive.
maybe though. So I would suggest that the true chronic, like these types of chronic eosinophilias are probably rare, but I'm not sure that people who present necessarily with just an acute eosinophilia would be all that rare. With an infection. So the question would be, how high would their eosinophils get, and is that maybe, if we were just trying to get people to submit a blood tube with high eosinophils from a technical perspective, like, can we actually get good quality DNA? Is that as difficult? Because I agree with you, true cases of like genetic eosinophilia, associated with a myeloid lymphoid neoplasm are going to be super rare.
And rip-roaring eosinophilia from a reactive cause is also quite rare. So we'll see it, and that's a good point, is we can ask our hematology lab to perhaps keep an eye out for it. But like the level that we are seeing failures with is not a common reactive level. Like if people have vasculitis or some other unusual cause for eosinophilia, which again isn't as uncommon as these myeloid neoplasms, we might see it and we might get enough of them to make that test, but that's going to be peripheral blood. The bone marrow is where they're really concentrated, and that seems to be where I'd worry more about it when you're seeing this half your cells are eosinophils and this specimen is so cellular, right? So, but that's a good point, is we can ask our heme lab to maybe flag cases with higher eosinophilia for us, maybe, I don't know, 4 or 5 or above, which isn't common, but not vanishingly rare, and see what it does.
Yeah, yeah. So, anyways, and we need to figure out either taking the chemical approach like PMSF for try to inhibit the proteins, or to try some other kinds of extraction methods. Hubert, did you have a comment?
No, I think it's a great utilization question to address also, right? Who should be allowed to work up, potentially trivial eosinophilia, and what's the best approach there? Is that really the best utilization of genomic resources? I think if they progress to the point of actually doing a bone marrow, as Eric just kind of mentioned, it would be nice to make sure that, things have been worked up. Specifically, if the primary query is, why is there all these funny eosinophils around?
I know we
I
used
think
to at UHM, we used to test a ton of spurious eosinophilia cases. We didn't triage them very much, but I think you're probably right that it would be better to have guidelines, really, that would stop unnecessary genetic testing for cases that clearly didn't have, that were probably just reactive.
this is
Right.
where, to have some other kind of layered, redundancies, whether you do still have to have some FISH or RNA fusion panels on your NGS when OGM fails. I think the issue is when OGM fails and then you start additional testing, especially for more acute cases, that's always kind of been problematic.
So that's interesting to me, though. So, like, are you getting results? Like, if you get an OGM fail, are you able to get DNA to do an NGS panel? Because it would still kill it. It's still digesting the DNA the same for both inputs, right?
So usually don't have a QC problem on the DNA RNA side for these cases. So I think it's specifically something
Or the
when OGM
long.
fails? Yeah, is it because of you're still getting DNA, but you're not really getting really long sequences? Um,
You know what would be really interesting to do actually is to run these failed DNA extractions on the tape station or some kind of, long, like where we could actually look at the sizing of what gets extracted.
Yeah, not a bad point, Adam. I think the DNA is probably, if it's there, might still be quite long, but we could definitely try that.
Yeah. I mean, again, just trying to eliminate, because if it works for sequencing, right, you probably got 1,000 base pair or longer fragments. If it's not working but I mean, again, Eric, you said the DNA yield was so, if you have the same amount of input, the DNA yield is less. When you're measuring yield, you're not measuring length at that point. You're just measuring, basically your like qubit reading, right? So there's clearly a lot of destruction that's going on. Anyways, okay, great. Well, I think that's was great. I think great discussion, some good ideas. If anybody has any good ideas, maybe how we can work together to continue to solve that problem collaboratively, because again, as Eric said, this is one of those situations where these don't real the real case these real cases don't come up very often, a couple of times a year probably. So need to we can definitely work together on that if there's anybody that has interest in that. Okay, so at this point, I think we can go back to the agenda item, which is the open discussion. I know we did have a fair amount of open discussion already around sort of the Stratus Sapphire chip and reagent problems. Just to go back to those topics, I think we have sort of talked about. Eric, you're still sharing, by the way. Just, just in case.
Thank you. Just responding to someone right now in this chat, so it's perfect. I'll stop sharing so you don't see that response.
All
Funny,
right,
I always
there we
people do that. Go, oh, I wouldn't do that. I did that. Thank
go. What was I going to say? Oh yeah. So there are a couple of points I don't think that we've got to yet in the open discussion. DNA extraction automation, turnaround time for urgent cases, limitation of ploidy small clones. I think sort of the viability of Stratus is one of the other points. I think we've talked about that to some extent. I think that's a bit of an open question at the moment because I think people who have newer Stratuses maybe aren't having the same problems people who had the original gen Stratuses have had. And maybe if that's your case, you should just ask for a replacement Stratus with a new one. But anyways, I think what we should do, I'm happy to go back to any of the topics that we've discussed already if there are points there, but I would like to open up. I think the sort of the automation questions and turnaround time for urgent cases are a couple of interesting points. So I'd like to just open the discussion up there for anything in the DNA extraction automation turnaround time. If anybody is using any automated, I mean, there really are a few limited option limited options for automated DNA extraction. I believe the Hamilton long string is the one option, and then the only other one I'm aware of is the Purigen Ionic, which is not available yet, but probably 2026 I think is what Bionano is projecting for that. So those are the options. Purigen Ionic I think will be first deployed for extraction only. And Hamilton Longstring can do both extraction and labeling with a DEC modification. So I don't know, is there anybody actually that has tried any of these? Oh yeah. I know, Eric. I know. They promise and never deliver is has been the yeah. for Ionic. I do believe they have a working. I know. I don't know if anybody saw Connie's presentation in September. I think it was one of the BioNano sponsored presentations. Connie Neveling from Radboud in the Netherlands. Um, they presented some preliminary pilot data on an alpha ionic that they had been working with BioNano, and some very interesting, very promising, I think, initial data from the Purigen. The one really nice thing, I don't know if I didn't know this, and so I will say it to everybody, is because it also touches very, very briefly on the turnaround time issue. The one really nice thing about the Purigen extractor that I did not appreciate was that because of the method, the isotachophoresis is all in liquid phase. So unlike the current DNA extraction methodology where you bind to a disk and you pull the ultra-high molecular weight DNA out of solution and you precipitate it, and then you have to rehomogenize it back into elution buffer, the isotachophoresis never actually is fully precipitated, and you just actually pull it out. It's in liquid phase. So you can literally go from extraction to labeling in an hour after the instrument has run. So there's no more 24 to 48 hour homogenization window. So that's a considerable, when you think about that, that is a considerable reduction in the processing time from enters in the lab, signs out report for that particular method. So again, I think that's very promising. I agree with you, Eric. It's like it's better, like it needs to be yesterday. Like we need access to these instruments, but yeah. So that was one of the interesting things I learned. Is it anybody has any comments? Anybody thinking about getting a Hamilton or automating this? No? I
Yeah,
know.
we've been looking at the Ionic and had it ready to ready to go, and then they kind of said no. So we're very excited for it, but haven't considered alternatives.
Yeah, I think the nice thing about the Ionic, and again, I don't want to speak out of turn because I don't have one in my hands, so we don't know what it's going to be until it actually gets released. I think the nice thing is the instrument itself is not really, from a CapEx perspective, is not super expensive. And the reagents themselves are actually not very expensive. So like, and I might be wrong. I don't know where eventually it's going to land commercially, but my understanding was that it's almost cheaper to do run ionic than the manual extraction from a reagents perspective. So if that is true, that holds, that would be really, really promising. The other thing is, so the Ionic has the capability to do 8 extractions in about 3.5 hours, I believe is the window. Hamilton can do up to 12 and in the same window. So 12 is pretty much the maximum that the deck can handle in terms of parallel processing. Of course, it's fully automated, so depending on how you set this up, you can load up. I mean, you could theoretically load up a Hamilton to run 3 times a day if you needed that kind of capacity. But yeah. Okay. I guess nobody's crossed the automation threshold just yet. So anyways, I think we are going to move in that direction. And so when we have some more experience with it, I'll come back to this group and present on how things went. And I myself, or maybe one of my group that's on the technical side that has more experience actually touching samples than I do. Okay. I will move us back to any other topics on the open discussion. Again, reagent fits, chip quality concerns, technical support, report automation and scaling was one of the other topics that we haven't touched on yet, really. So if there's any other comments or questions or topics for discussion, now is the time to bring them up. Pretty quiet out there.
Yeah, maybe I'll bring one topic that I know you kind of addressed, Adam, in one of your recent papers was some clinicians here at Sunnybrook were wondering if OGM reporting could be separated into these are the things that what you really expect to see on a karyotype, and these are all the extra things that OGM is revealing. So this comes up really because our clinicians are already very busy, and sometimes they just want to plug things in to certain clinical risk calculators, and they always get mixed up. Should I be including this or not? Does this count or not? So I don't know if anyone else has received this question before.
Yeah, that's actually a really interesting point. Tara, over to you.
I would say, at least for our acutes, we're still performing karyotype analysis in tandem for all because we're issuing those results within a couple of days typically. So at least for those indications, we already have the karyotype to correlate. And I do sometimes include a point under the ISCN that says number of cytogenetically visible abnormalities. I don't always do that, but occasionally I do. And I have had those questions by email to clarify, are these also karyotypically visible or not? So I think it's a good point to discuss for sure.
Okay, something else to work on with the BC team there. So again,
well, I think it's an interesting problem because the way the framework guidelines for reporting were thought about were really from the context of recapitulating a karyotype analysis. But you are correct in that there are some things that are just not visible by karyotype Nicholas's presentation today gave several good examples, of stuff that, and Eric, yours as well, actually, of stuff that wasn't, not everything visible by karyotype. So how do we differentiate that? I guess the problem is that at one point, once you get over 3 or 4 abnormalities, I'm not really sure that we're changing prognosis significantly. And if inside the mess of a complex karyotype, you have a targetable abnormality that has then I'm not sure again where the clinical, where the calculator maybe be so relevant in that case. So we might be actually just talking about a subset of cases that require clarification, if I'm understanding that correctly, or maybe it's more for MDS than it is for AML. Eric?
Yeah, like Tara said in the chat, it's the ones that are borderline that are where we think more about it. So if we're seeing like 4 abnormalities by OGM, we start questioning which ones can you actually see by karyotype. Because I think, like someone said earlier, the complex ones, or that was a different call, sorry. The complex cases are almost never a question, like they're chromothriptic and there's just chaos. But yeah, it's complex, great. But the ones that are like complex because they have 3 abnormalities by OGM, that's kind of where we question, are they really complex? And we'll more often reflex a karyotype if there isn't one already for MDS, for example. We do try and provide an IPSSR score for the cytogenetics at least. So if it's 1 abnormality, great. If it's 2, still okay. If we're in that range of it's becoming complex, but it's borderline with OGM, then is it just the increased resolving power we have now?
Well,
So we tend to reflect that.
it's the fundamental epistemic problem of operational definitions.
Yeah.
Right. So we operationally define a complex karyotype as 3 and above, knowing that that's a bucket of muddy water. Like if we were to separate it out, I'm sure there's cases that theoretically fall into the complex bucket that probably aren't necessarily, that bad prognosis, and others that are. And so OGM just further muddies the water by allowing us to see more cases that put a few more people into that category probably. So yeah, it's certainly a definitional issue. It's a problem with any operational definition, especially operational definitions for chromothripsis, for example, are very problematic. Cells can't count. They don't know how many, segment abnormalities they have. They don't care. They just form micronuclei and break into lots of little pieces. So, this is the problem with approaching any of these problems with an operational kind of definition. So, yeah, but I think that's a good solution. What you guys are doing in the borderline cases, just to say like, hey, by the way, this is borderline. We think this is visible. I myself would sort of err on the side that even if I had a translocation that was cryptic, but I would still count it and say, hey, like we wouldn't have seen this by karyotype, but the underlying idea of the karyotype rule is that once you hit that 3, you've got enough sort of genome instability that you're probably in complex territory. So I don't know what you think about that, but
That's my usual approach as well. The scary corollary is if you're not doing karyotypes well, you have the risk of missing those things that grow in culture that would have counted before, which we've seen as well, but risks to everything. So I wanted to point out or plug that the ICOGM group is also working with a complex karyotype subgroup on trying to operationally define what is it in OGM and what matters in OGM. So I'm sure it'll take a while because it will take a lot of data that is complex to try and make something of it, but try and get a better answer for what number what number matters in OGM.
yeah. And it may not be the
What-
same for every disease. I think
yeah.
I mean, we've sort of set different limits. I mean, in BALL, the limit for complexity is 5. And CLL, I believe, is also 5 for karyotype, but for other modalities could be different. Yeah, so I think there's a fair bit of complexity. It was certainly nice to think 3 or more across the board, but I think that's maybe overly simplifying the biology. Um, Anyways, yeah, and thanks, Eric, as well for the plug for the ICOGM. If anybody's not a member of ICOGM and you want to be a part of it, it is really getting started again. But we are meeting more or less monthly. You can go to icogm.org and register to be part of the group.
Yeah, I was just going to add, like, it's not necessarily like, large things that OGM can pick up, but even some things like loss of heterozygosity, which the data is changing all the time. So even in something like in NPM1 AMLs, or again, whether you have a double hit or just one mutation and loss of heterozygosity on the other allele, also starting to show that those are actually worse prognosis and worse overall survival. So again, the question of what's relevant now, what's really relevant moving forward, even from year to year, such a moving target, right? So I'm just going back to that question of, would you see this by karyotype or not? As is it relevant or not? Well, it may not be relevant in this old school risk scoring system that they used because they couldn't see it anyway. But actually, right now, it actually is relevant. So,
yeah, no, totally, and I think that we're in this phase. We're in this phase now where we're actually we have the tools to actually get this data in high resolution. So it's the pace at which we can sort of start to make these kind of correlations is improving. Okay. Well, it is, what is it? It's 1:41. We're coming close to the end of our 2-hour window. If there's no other comments or questions, we can bring things to a close. I will just point everybody, very quick reminder that Jenny Bozek put in the chat the link to the evaluation for this community of interest event. So please fill in the evaluation, and that's how you get your certificate of attendance as well. And I'll also just mention very briefly that this session was recorded and will be sent around. There'll be a link sent around later, but you can always find all of our COI presentations on the LMP website. So if you just Google OGM Community of Interest LMP or something like that, you'll find the link to all the past recordings. Any other final comments in closing? Eric, Susan, anything to add in closing?
Thanks everyone for a great discussion. I'll send out that listserv again after this meeting, as well as the survey results as requested, and a way to maybe touch base on who may be interested in eosinophilia and how we can evaluate that as a group.
I was just going to say that was a great plug from Eric about the listserv, and that Adam's point about sending cases to spark and stimulate another meeting in a more timely fashion is a really great suggestion. So yeah, both of those.
And don't forget, if you're having reagent issues, and you want to sort of compare notes with people if they're having a lot, like let's reach out, let's figure out a way to do that so that that we can put our efforts together, and if reagent lots need to be replaced more promptly by BioNano, we can work together to make that happen. Great. Okay. Well, I want to thank everybody for their participation today. It was a great meeting. These are always fun to have, a little less formal than a conference, and it gives us a real opportunity to have discussion, which I think is really important for us all to develop our experience and knowledge with OGM. And it's clear. I want to thank Hubert as well for also bringing forward all the sort of the clinical points and the work you're doing with CLSG to sort of just to make sure that people understand how important clinically OGM is. Yeah. Thanks again. We will try to have another one of these in 2 or 3 months or so, hopefully. We'll try to keep the momentum going, keep everybody talking, and thank everybody for being here today.
Thank you, everybody.